Hello everyone, I hope you can all hear me well. Welcome to the latest Ontotext webinar. The topic today is AI-powered target discovery with speakers Ilan Uzunov and Martina Markova. They will present themselves in a few minutes, but before that, I'd like to point your attention towards a couple of housekeeping details that are important, and we will wait for some additional people to join as well. So first and foremost, you are all currently on mute, and this is to avoid background noises and to enable the speakers to present without any interruptions. However, you can still communicate with us using the question box in your GoToWebinar panel on the right, and we actually strongly encourage you to do that so we can have a meaningful conversation at the end, because we have planned this Q&A session, which will be around 10 to 15 minutes, depending on the number of questions that we've got. Other than that, the session is being recorded, and you will be able to access the recording most probably tomorrow. I will send it via follow-up email, so you will have that alongside the slides, and also any questions that we might not have the time to answer today. We will also make sure to include them in this email. So with that, I would like to now give the floor to Ilan, who will start the actual presentation. Ilan?
Thank you, Yasin. Just to double check the sound, can you hear me well?
Yeah.
Thank you. I have the pleasure to be accompanied today with my colleague Martina Markova, who is a senior business analyst in our life sciences solutions team. As you know, we have a dedicated team of subject matter experts that have a really deep understanding of the use cases our customers are facing. And my name is Ilan Uzunov, and I'm heading sales and business operations in the life sciences and healthcare industry verticals, which are quite central for our company. Without further ado, let me present actually what we'll be covering today in terms of topics. I'll do a really quick introduction of Ontotext. Then we will discuss some of the main challenges biotech and pharma companies are having in their drug R&D journeys. Afterwards, we will have some time to share with you the specific AI-powered solution for intelligent target selection that we have developed. Of course, we will share one case study from an active customer of ours in this space. And of course, we have left some time for a demo. I wanted also to share with you that we have prepared a short poll. So 2 questions will pop up at some point. So please feel free to share your feedback. This is important for all of us. And of course, the results will be visible for all users. All right, then let's kick it off. So some basic facts about Ontotext for those of you who haven't heard about us. So Ontotext is one of the first semantic technology companies that appeared way back in the year 2000. So I need to fix this slide for sure. We're turning 22 this year. And we're entirely focused around 2 main capabilities. One has to do with working with huge volumes of dynamic and diverse data, both structured and unstructured, and consolidating those data points in huge holistic knowledge graphs that address specific use cases for our customers. And the other big capability that we have is that we additionally enrich those knowledge graphs using important insights and relations that we extract from documents or from unstructured data. We're doing so to help our customers actually to solve a lot of problems, starting with discoverability or more intelligent decision support or deeper analytical capabilities. We're a global company with sales and operations across the globe. We have presence in New York. We have office in Basel, another presence in London, and the headquarters where all engineers sit is currently based in Sofia, Bulgaria. And we are happy to serve a high-profile list of customers ranging from leading science, technology, and medicine publishers to leading and global pharma companies, as well as leading hospitals across the globe. We have deployment scenarios on all major cloud vendors, but also on-prem. So what do we do in a nutshell? So we believe that we help our customers to actually develop a really powerful data assets that we call knowledge graphs. And why reusable is so important here on this slide, because that we believe that knowledge graphs could actually evolve continuously with the time and become one of the main assets for the company, because they are supposed to be able to provide deeper analytical insights on top of your structured and unstructured data. So very often we work with our customers' proprietary data, but we very often also enrich it with relevant public data sets. So why knowledge graphs? Why there is such a big hype around knowledge graphs? Let me tell you my story for that. Knowledge graphs, first and foremost, give you a totally new perspective for managing and consuming data. So they help you with the transformation from having to work with isolated, siloed data stores that keep specific data points to being able to navigate through your data based on your specific business needs and use cases. So the data is described in terms of the relationships between the concepts, and you can actually trace them down based on the requirements of your business people. So for instance, if you are in drug R&D, you will be able to trace down the relationships between your drug candidate and the specific protein that they are targeting. You can even go on a very lower granular basis and check additional data, like for instance, gene expression data, molecular interactions, and many, many others for your specific therapeutic area research teams. Last but not least, graph databases and knowledge graphs could be used in many different ways. They could be used as traditional databases that could be queried. They could be used as graphs that you can run various network analytics on top. And last but not least, they could be used as reference databases on top of which you can generate new facts and knowledge. Let me now share with you one of the common challenges that have serious business impact on biotech and pharma companies in their drug R&D journeys. First and foremost, they have huge issues when it comes to deriving key findings so that they can confirm specific molecules, targets, or compounds. This has to do with specific therapeutic areas, of course, and the research teams that are working across the globe are having huge issues because they're mainly doing this in a manual manner. And very often they are not leveraging data-driven ways to generate insights. In direct relation to that, this can translate to actually increased backlog of compounds, therapeutic targets that needs to be additionally screened and validated before they go to the labs for the actual research. Very often this also leads to declining success rates with new molecules, and something which is really even more hazardous in terms of impact on the overall drug R&D workflows is that they may even lack efficacy in respect to the specific intended indication for the specific disease that they are trying to treat. At the end of the day, all those translate in exceeding research budget and loss of competitive edge because the inability to effectively collaborate insights across the R&D functions of global companies translates into increased inefficiency in the overall drug discovery processes. And this has tremendous impact on the way that the organizations is actually being present on the market. This has direct impact on their competitiveness, if you will. So let me try to relate some specific technical issues that actually correspond to the critical business issues that I addressed in the previous slides. Very often, research teams in their day-to-day work need to handle huge volumes of public life sciences data. Those are very often scattered, isolated, non-connected data stores that they need to go and work with on a daily basis, extract insights which they believe are relevant for their research, put them in an additional application. Very often this is Excel or Word, so that they can use it for their continuous research. And this takes a lot of time and effort. The other huge problem is that very often the data is inaccessible because the data is actually hidden in unstructured data sources, very, very often in documents or research articles or in patents. And this poses another complication when it comes to the specific research workflows that needs to be followed. Once again, the researchers are extracting all those relevant findings from PubMed or from medRxiv or for patents data, and they use it for their research. Last but not least, another huge problem is that data is very often unreliable. And this is because the metadata is of real bad quality. It's inaccurate, and the researcher cannot actually be sure that he or she can use the specific finding that has been generated or has been discovered in a specific source of information for their research. And this can lead to good results for the overall organization. So how we believe that we address those technical issues that have direct impact on the critical business issues in general. Of course, on the first side, we believe that we help our customers to actually generate deeper insights on top of interconnected knowledge network, which very often consists of huge volumes of facts, as you will see in a minute from the case study that I'm going to present. And they are able to trace down long sequences of relationships between the different biological entities that they're researching. On the second place, we're also facilitating information discovery on top of siloed data, which is actually interconnected. If you remember what I said in the beginning, that very often in their day-to-day research, scientists need to work with different siloed data stores. Now with our solutions, they're able to access everything from a single interface, which is really makes their lives easier. We also believe that we have direct impact on the innovation, and we can actually drive innovation at scale, because the way our solution is designed allows the data model to continuously be adapted when it comes to the specific needs of the different use cases our customers are addressing. Of course, this also translates into lowering costs not only for the researchers and the way that they work, but also lowering costs for working with data, adding new data, maintaining and updating data. And of course, last but not least, we have put a lot of effort to make sure that the data is of really high quality. We have specific metrics like confidence scores or provenance that would allow us and our customers to trace down the specific insight and the source from which it originated. I would pause now and ask my colleague to launch the first question from our poll so that we can have some early interaction with our attendees. So quickly, if you are able to select one of the options. So how easy is currently for you to derive new therapeutic target insights and findings from your data? Please select one of the options. Those are, we're struggling with finding new targets. The backlog is too big to test new targets in a timely manner, and we are able to consistently expand our pipeline. I'll give you 5 more seconds, and then I'll ask my colleague to show you the results. Okay, yes, and I think we're good to go. Okay, interesting split of your answers. Of course, we expected to see the first one. So 36% of you are struggling with finding new targets. And as I said earlier, one of the major issues is that the backlog is really big and there is simply not enough time and resources to go and manually validate all the target candidates so that you can actually end up by a good number of top priority candidates that you can go to the lab. And then, of course, a small percentage are able to consistently expand our pipeline. Okay, I think we're good. Let's move onwards then. I'll share my screen once again. I'll go to, okay. I hope you can still see my screen.
Yeah.
All right then.
We can.
Thank you, Yasin. All right, so as I told you earlier, the solution that we have developed covers specific needs for the target discovery workflows, and more specifically, it deals with target selection and target ranking so that the researchers can go to the lab with a really good number of target candidates which have been validated using different data-driven approaches, and they're also backed up by significant amount of scientific data, both structured and unstructured. So we believe that with this solution, we can bring specific benefits to the table for biotech and pharma companies, starting, of course, with the central one that we believe that we can help them to reach the market earlier and gain a competitive edge against their competitors. For pharma companies, we believe that we can help them to find new applications for their existing drugs so that they can also generate additional revenue streams for the company. On the operational efficiency level, we believe that we can empower our customers, computational biologist teams, data scientist teams, data analysts, so that they can be more efficient in their day-to-day work. And of course, last but not least, this translates also to a lot of cost optimizations in terms of not only resources, but time for your valuable subject matter experts. Our findings so far, working with our customers with our solution, shows that all those benefits have the potential to translate to specific gains ranging from tens to hundreds of millions of dollars per specific project. All right then, let me share you a customer case study from one of our active customers in this space. So before working with us, this biotech company have followed all the traditional routes when it comes to target selection and target validation. They were having their researchers using big number of public data sources, as I said earlier, many of those non-curated, non-connected to each other. So they needed to manually gather the data, analyze the data, process the data, align it with their colleagues, try to collaborate across the R&D function. And this translated to a lot of overheads in terms of resources, both in terms of people involved, but also time. So very often in their research, only for a single target candidate, they spent from several hours to several days. So when they started working with our solution, which provides access to more than 5 billion facts using 35 curated data sets and additional 100 semantic mappings between those data sets, our customers were able to actually work much more efficiently, leveraging the interconnected knowledge graph and exploring the hidden relationships within this graph as per their specific research and use case. Additionally, we added through natural language processing 40 million documents, mainly scientific and research literature from PubMed and MedRxiv, but also patents. An additional 500 million relations were extracted and were added to the knowledge graph. And in this way, we were able to help our customers to actually unlock new insights, new findings that otherwise were simply not easily accessible. And we believe that in this, we are now able to help our customers to deliver much more insightful information about any biological entity of interest to them. And this translates, of course, in terms of cost optimizations, but also time optimizations and access to market. This translates into huge reduction in the times that they spend to research a single target from several days, as I said earlier, to just a couple of minutes. Okay, I think now it's good time to also share the second question from the poll. So Yasin, please broadcast the question and let's have our audience that. Thank you. So we would be interested to learn whether you have some experience in your organization with AI tools, knowledge graphs, platforms of any kind that are used for data collection, data integration, or data analytics. So please select one of the following options. No, we are not planning anything like that currently. No, we are researching such AI tools now. We are in the process of adopting AI tools for drug discovery currently, or yes, the last one, we are experienced in using AI tools for drug discovery. I'll leave you 5 more seconds to decide which one fits best your organizational profile and current state of the art. And I think now we are good to show the results, please, Yasin. Okay, interesting results. To be honest, I wasn't expecting such a big number of customers, attendees that have already some good experience using AI tools. So 20%, almost 20% say that. Another 26% say that we are currently in the process of adopting such tools. And but still 44% says that no, we are researching such tools currently, which is good. It simply gives the overall perspective of how actually AI tools are gaining momentum in biotechs and pharma companies. Of course, they need to prove first that they can deliver value for the customers. So I understand that this takes time. So the more successful projects there are around the globe, and the more people are sharing information about those, the better. Okay, let me quickly walk you through the some of the insights. So what is actually under the hood? So I'll only speak about the data sources that we use. And here you can see some of them. I refer to those 30 plus data sources. As you can see, most of those are very well known reference ontologies, terminologies, or scientific literature sources for you guys. We have categories covering genes like NCBI genes or UniProt. We also have information extracted from Campbell and Drug Central about drugs and compounds. For pathways, we use all those 3 data sources, but we also do a lot of additional semantic mappings between the data sets using all those main classes of and entities that you see here on the screen. For clinical trials, of course, we use the source, clinicaltrials.gov. For scientific literature, we work with PubMed and medRxiv, and of course, for patents, currently we work with Google Patents. With that, I would like to hand over to my colleague Martina to walk you through the demo, and I want to remind you that we want to allocate some good time for Q&A, so don't be shy, share your questions in the Q&A box, and we'll try to address them. Over to you, Martina. Can you please check your audio, because I don't think I hear you. I don't know whether this is only with me. Yasin, please confirm. Do we have audio connection with Martina?
It seems like, but no, with Martina, we don't hear you well at this point. Can you check your? Sorry for the technical issue. We'll try to fix it in a few minutes or less, I hope.
Martina, you can also use your built-in laptop mic and headset if your headset is not working properly. I believe this could also be an option.
Does it work now?
Yeah, I think it's fine now.
Okay. Thanks for the understanding. So, okay, let's kick it off. We have the target discovery demo, and this is going to be a live demo. We have, what we see here is a fully configurable homepage that we have just used to showcase what options we have and how to configure the system. We have a couple of modules sitting on top in this top ribbon. Some of them, like the first 4, are used to find information about specific targets and find insights about them. And the last one, rank search, is used to help select a target among a list of possible candidates. And then we have a management module from where all of the system can be fully configured. Today, we're going to focus on some of these modules, so semantic search, graph path search, pattern search, and rank search. However, if you do want to learn more about the system, please feel free to contact us for a more in-depth and personalized demo. So kicking it off with the semantic search, this is a module that is planned to be used by researchers on a day-to-day basis. As my colleague already mentioned, we have various types of data sets that we can already use in the system. They're already mapped in a common model, and it's basically a plug-and-play principle. In this demo, we have integrated only a couple of them, but if you're interested in some that you don't see today, please also make sure to get in touch. Today's example that I'm going to provide is about a disease that still doesn't have a cure. It's the worst type of brain cancer called glioblastoma multiforme. And up to date, most patients that get diagnosed can live only up to 14 months after the diagnosis. So researchers are actively looking for new cures. The search can be performed in various ways. First and foremost, we usually begin by selecting an entity that we would like to search. I'm just going to give you an example with the gene. And I'm going to type in the name of a gene that's mutated in many patients with this type of cancer. And when I fill out the name of MGMT, that's the gene symbol, we get a couple of suggestions in the autofill. Maybe most of you who are already familiar with graph technology would recognize that these are actual entities. So all of them are objects that we have in the database. I might choose one of them, but I also might perform the search just by leaving MGMT as a string. So when I press the search button, the search is going to provide me suggestions about genes that have a name that matches the string. Clicking on the gene would give me the results table, and I see 2 matches. Why 2? Because we have 1 human gene and 1 mouse gene. If I get a longer list of suggestions, I can always use the filters to narrow it down. All of these filters are configurable, so we can add any information to them anytime. We can add, for example, organism as well. In a related disease, we can already select some abnormalities and check the gene that comes forward in a specific disease. Now, clicking on this entity opens a new dashboard. Because I want to show you something else as well, I'm going to open it in a new tab and get back to the search later on. And here we see the dashboard of the gene, which gives us a 360-degree view over the specific entity. Now, we have dashboards that are also fully configurable with different widgets. And currently, the system works like that. We have an entity called gene, and the dashboard relates to this entity. If we go to a protein, which I'm going to show you after that, we will see a different dashboard. So all of these dashboards can be tailored specifically to the information we require about a specific entity. We have all sorts of widgets here. Some of them display only text, such as the official gene symbol or the full name, gene type. We have some that look more like plots, such as the publications per year. Here we can see a peak in research of this gene around the early 2000s, like 2008 to 2014. And now we see a fast decline. And we also see interactions, interacting partners. And you might notice that even though we are using NCBI as a main source for gene information, as my colleague already mentioned, we also have various other data sources which are all seamlessly integrated in this dashboard. So we have interaction data coming from BioGrid or StringDB. We have publications coming from PubMed. We have integrated also Ensemble. So there are literally no restrictions in which data sources you can use. You can integrate all of them. We even have variants coming from CleanVar. Now the related protein, I'm just quickly going to show you what the protein dashboard looks like. I'm also opening it, so I don't need to go back to the search for the protein. I can directly open the protein dashboard from here. And you see that I already see a tailored dashboard that shows me different information. This time I can even go to compounds associated with proteins or even associated drugs. And these two come from other data sources such as Kimball, respectively Drug Central. Now, before I move on to the next type of search, I just wanted to provide some information on the NLP that we're using. This is one of the interesting features. Just keep in mind that I have typed in here a simple string, MGMT. I can also search for the string in PubMed directly. And here I get a list of articles. Currently, for this example, we have only loaded the abstracts of the articles, but it's possible also to load the full text of these articles. And again, I see a list of results that I can filter down by publication type, descriptors, and I also have a dashboard. So open it in a new tab. And we see here a document widget. So this is specifically made for text information. And we already see how the NLP has done its job in recognizing MGMT as a gene, for example. We see methylation, that's a function. And then we see protein, glioblastomas, which is a type disease. We can also have another look at this by looking at annotation types. We see that in this text we have 5 genes, 5 diseases. And we can also check what's the annotation score for this. So we see glioblastoma has been recognized with 100% confidence. If I go down, I see the protein expression, for example, is not anymore marked. But this is an easy way to see how has the NLP done its job. And then I have the mentions, so I can see what has been mentioned in this document. So the whole information from these articles has been seamlessly integrated in the graph. And we don't really need to go to read the full article, but we can just skim through the abstract and then decide which is the most relevant for us. Now, I just showed you a type of search where we type in the name of an entity that we're interested in, and we get sort of a holistic view over this entity. Now, I'm going to show you the graph path search next, which is meant to help me identify new facts. All of these facts might come from different databases. They might come from NLP, or they might also be inferred, meaning that we combine different data sets in the graph, and the graph then automatically predicts new properties or new relationships of these entities and makes new connections between the dots so that I can easily make insights about new information without searching every database one by one. So the way it works is that I have to choose sort of a mode. We have the shortest path. We can find all paths, distance between nodes, and so on. For the sake of time, I'm just going to show you the shortest path search today. Let's make a search between the gene that I just showed you and the first line of therapy that we have for the worst type of brain cancer. I'm going to choose the drug as the second entity. And the first line of treatment for glioblastoma is called temozolomide. And it's currently the only drug that kind of acts. All of the other therapies are more like adjuvant therapy. So currently the only weapon we have is temozolomide. And unfortunately, whenever MGMT has a mutation, then this type of therapy doesn't work. Mutation is not exact, so for those of you who are researchers, it's called methylation, is the right term. And I can also add filters. So down below, I just added mentions because whenever we search for the shortest path, usually the results will show me which article mentions both of these terms. In this case, however, we are not interested in the articles that are mentioning them, but in the other nodes in between. So we can find out more about which diseases both of these come in, or what other therapies might be there that target MGMT apart from temozolomide. Okay, let's hit the search button. Now we have started a search over the whole graph, which currently contains about 5 billion facts. Now, before a webinar, I also executed the search to get the results so that we can be a bit faster with the time and have some more time to answer questions. So this is what the result would look like. We have here the gene, we have here the destination node, also the drug, and this is what the result looked like. These are actual nodes. On one hand side, we have the gene, here we have the drug, and these are all the terms in between. So we see a couple of diseases like colorectal carcinoma, neoplasms, we see some cells in between, and interestingly, we see also other drugs. So in case we want to find out, okay, what other drugs are there that target MGMT, here is our answer. We have fluorouracil, doxorubicin, dinoprostone. In case I'm not sure what one of these entities means, when I click it, I will see an overview of the information that I have from the source. So in our case, we have Drug Central as a source for drugs, and I can see all of the information that this database actually includes. Now, if I'm not sure which of these would be most interesting to me, I can also let the graph guide me a bit. We have a heat map tool that, when pressed, will cover all of the entities in the graph. And by clicking the color legend here, I can see what does it mean. We have some rankings. We have rank 76 for fluorouracil. We have rank 74 for MGMT. This rank comes from an algorithm that works on the graph, and it evaluates how important is a node in the current context. So it can literally tell me fluorouracil is the best connected one. It has many relationships to other objects, so it might be important. I can also check how does it relate to other objects by clicking the predicate. This is also known as the relationship between the nodes. So we see link structure. Okay, we need a bit of knowledge of the data model, but in terms of Drug Central, this is how they tell us, okay, this is a related structure. So these two have something in common from a chemical point of view. We also see that disease is related to fluorouracil as well. It responds to the therapy, and fluorouracil can be treated, can be used to treat colorectal carcinoma. Now, if I find that this is one candidate that I might be interested in, I can also go on and explore it further by clicking the entity view. The entity view loads sort of a table that provides an overview of what do we have in the graph. This is especially useful to data scientists who would like to build, for example, new dashboards. If I'm a researcher, I might be more interested in opening the dashboard directly. So let's go to the dashboard for fluorouracil. And here I see the drug dashboard, and I can also jump to the compound, for example. I can see most common adverse events with probability, or also indications with highest clinical development phase. Of course, the lower the phase, probably the higher the chance I will have to explore this drug further and to make it my next successful candidate. Okay, so we saw two types of search. I think it's time to see also the third one, which is called pattern search. This type of search is usually configured by the data scientists, and it is used to identify properties that are common for certain objects. What does it mean? We have here templates that we have built beforehand. Both of them can be used to identify different things. The first one can give me all genes and proteins which are targeted in a certain disease by a specific drug. The second one, which I think will be more interesting, is to find all drugs which target a specific gene or protein in a disease. Let's see how it works. I'm going to select this one. And here we see that we have two fields. We have a field where we type in the disease, and then we type in the gene. And what we would expect as output is a list of drugs that target the gene in a certain context, the context of a certain disease. Now, one thing that is worth mentioning is that even though so far I was talking about the worst type of brain cancer, we're not restricted in any way to a specific therapeutic area. You can equally well search for other types of cancer or for other diseases as well. I'm just going to show you, you don't even need to know the exact name of the disease. You might also type in something that's more like a global term like polyps. That's just a specific type of tumors. And let's type in here the G symbol of kinase that's very often mutated in cancer. And when I hit search, I get the drug and I get the mechanism of action. And we see that all of these are inhibitors of this kinase in cancer. And likewise, as you saw before, we can go on to explore the drug dashboard for each of these candidates to learn more about it. Okay. We saw three ways in which we can search for potential interesting candidates. What might happen in the end of these searches is that the researcher ends up with a list of candidates. It might include 5, 10, or even hundreds of these. Usually, an organization will have some rules or criteria with which to decide which of these will be the most promising ones. And it can take days to determine which of these correspond to the criteria best. This is why we have built a tool to make this process more automatic. In this tool, we have pre-configured one criterion, which to be used to order the candidates that we have in the list. The criterion we're going to use is called importance. However, bear in mind that you might have more criteria and they might be organized even in hierarchical structures where you assign weights to them and where each of these criteria have a different impact on the final ranking. So what we do first is to start by choosing a file. In this file, I have already collected a couple of protein IDs. It's a CSV file, so really easy and quick to make. And then when I click on protein, I get to choose which of these proteins that I have in the file correspond to which identifiers from UniProt that we have in the graph. All of them are automatically matched. So I'm just going to click on select all, save the selection. This would also allow you to change some of the matching in case you're not happy with the way they are matched. And then I get a list with all of these proteins, all of them related in a way to cancer. To see which one has the highest importance, I just sort them and I see, okay, epidermal growth factor receptor is the most important in my list for cancer. It's kind of expected. We also see TP53 albumin, which is often used as a reference, and so on. We can also preview the ranking that we have currently configured. We see a top level called importance, and we see 4 levels underneath. So you can see that you can assign various categories and subcategories, all with their own unique weights. And in this way, you can get a really flexible, small algorithm in which to rank the list of potential candidates. Okay, we saw a couple of options for target identification and target selection. And now I think it would be best to summarize a little bit. So I'm going back to the slides just to show you a few takeaway messages. The first one, of course, is about successful target selection or identification. And we know how tedious it is for a scientific researcher to traverse through all of these data sets one by one. It can take days, as my colleague already showed, to just find out one that would be suitable. And we are empowering researchers to leverage public data sets and automatically extracted data from papers all in one place without having to change the system, and all with customizable options. Once the researcher has gathered a list with potential candidates, it might be really hard for them to decide which ones to invest in further lab research, which is why we are also supporting these critical decision-making processes by making automatic ranking and mitigating risk by leveraging provenance and evidence metrics. Evidence and provenance is one thing that we are actually providing on all levels of information. Today I showed you the annotation scores for NLP, but we also provide provenance and evidence for everything that we find. So we provide, for example, you saw BioGrid as a source for some of the interactions. We provide also scoring into, okay, how many data sets have we found, or what's the importance of all of these data sets that we're using, so that really when the decision has to be made, the researcher can be sure in what they have found out. And then the last point is perhaps also one of the more important ones. You saw that we have some features that are more suitable for researchers, but also for data scientists. And they can both leverage this technology, for example, NLP or knowledge graph, in a way that's more suitable for each of them, user-friendly and customizable. So the modules for researchers, they would mostly go to the search, like a semantic search, maybe pattern search, graph path search, while the data scientists might be more interested in exploring the database by directly querying it. This is the 4th module of search that I didn't show you today. It's called the Sparkle search. And we're making sort of a crossroad where both of these can meet and can collaborate successfully. With that, I would like to invite Ilyan to share a bit more about the next steps.
Thank you, Martina. I hope you can all hear me well. I just wanted, before going to the questions, we have some interesting questions, to invite you all, of course, all who are interested in signing up for a free workshop with our subject matter experts. We would like to hear more from you when it comes to your specific target discovery, target selection, where use cases, challenges that you're currently having, and discuss how potentially we could be of help with our relevant expertise. Okay, with that, I would like to spend some time for the questions. We do have several questions from the audience already shared. So one of the questions is related to actually what is our specific add-on, because this attendee says that Dimensions is great, Campbell is great, they're quite advanced. EBI gene ontology is also advancing, but what is your specific add-on? So here I can very quickly relate to our linked live data inventory. This is a data offering by Ontotext in which we spent and invested a lot of effort around not only transforming all important reference terminologies, ontologies, and data sources in RDF, but also introducing additional semantic mappings on top of them using all main classes of information that you saw already, like genes, proteins, diseases, drugs, you name it. And this leads overall to having a much more harmonized data in terms of output. Data which is interlinked in terms of concepts, but also data that will allow you to trace down provenance towards the specific data source from where a specific description of an entity or class of entity has been derived from. Okay, the next question is, how do you verify that the new relationships extracted through natural language processing are not noise? Here, I can share with you that we have confidence score calculated for the named entity recognition, which we propagate to the relation extraction as well. In addition, we have a whole set of tools, additional toolings that we use to create training corpora, to do data quality assurance and curation. We have a dedicated product which is called Ontotext Metadata Studio that could be used for that, and it's tightly integrated with GraphDB. And this allows us to not only create the specific training corpora, but also continuously monitor the quality of the NLP outputs. Okay, let me check whether we have some new questions. Yes. How do you know? Yes. How do you know how recent the data is? How often the data is updated? So maybe Martina, would you like to take this one?
Sure. So we do have incremental updates for PubMed articles, and for all other data sets, the customer can decide how often to update these. All of the data sets can come with their own specific upgrade frequency. I think for PubMed we have 6 month to 1 year or yearly updates, but they can also be done monthly depending on the requirements of the customer.
Okay, thank you. Another one from the audience, did Ontotext implemented weighted properties using RDF star? That's exactly the case. We are calculating like a global score for a specific edge, and on the basis of the sources that it's generated from. Okay, the next question is more or less the same. Are you using RDF star? Yes. How would we go about analyzing 50 billion or more nodes? As we already shared, the current case study that we shared with you runs on top of 5 billion facts. We have implementations which are way beyond that. So 50 billion is within the range that we are feeling comfortable working with. Is the full text of PubMed stored in the Graph Store or search engine index? Martina, can you help me with that, please?
Sure. So we do store the full text in another database called MongoDB, which is customized or tailored to storing text values. And from there, we also populate some of this data in indices.
Thank you. Then another one, how easy is for non-technical people like scientific personnel to leverage this solution in their everyday research? Martina, please, can you share your perspective here?
Sure. Sure. So usually the way it works is that the researcher would define their own, let's say, search criteria or search workflow, and they can either work together with a data scientist to make the customization that they're required, or they can also purchase a workshop that we can customize for them so that they can learn how to customize the system themselves. Or we also offer professional services where we work together with the scientists to meet their requirements for search, for visual exploration, such as dashboards, or anything else that they would require the system to be able to do according to their needs. And in terms of data scientists, we also have various options. They can either work also with the UI and completely customize the dashboards for themselves. They can also explore the data and query it directly by using an SQL-like language, which is really similar and easy to get going. And for them to get going, we also offer custom workshops so that they can leverage the data model and start using the data efficiently.
Thank you, Martina. Another one that just came in. Could you identify novel targets with your product rather than selecting for already validated targets in a disease?
I guess it will be also for me. Of course, the idea is that we can also identify novel targets, especially the graph path searches where we can find new insights or things that are still not covered. And by using the dashboard, you can customize it so that you can see, are there already any drugs that target this specific candidate? What stage are they in? Is there a compound that has been worked on? What does the literature says about it? You can also integrate information about patents, and that's how you can see how novel this target is. Yeah, there are also various other customizations that can be done so that you can also have a brief look at the target and see how novel it is. Also in the rank search, just another method to understand how novel a target is to define a criterion called novelty. Under this criterion, you can define various other properties so that the list of targets that you can identify will be ranked not by importance, like in the example that I just showed, but they can be also ranked according to their novelty.
Thank you. Another one, which is more broad, but I believe it's really important. Do you have a focus in a specific therapeutic area? I can handle this. You can do it for me. It doesn't matter. Maybe to do it quickly. No. Although we have quite a good experience in oncology and additional therapeutic areas, but actually the good thing about this solution is that it's universally applicable to different therapeutic areas, simply because we're leveraging the huge abundance of high quality reference data, but also scientific literature, patents, and all other relevant data sources we're using. Okay, any possibility to proceed to chemical structure search within the graph? Currently, no, but actually this is also a direction we're looking for. Currently, we have we can trace down the specific relationships between drugs, structures, and compounds. But yeah, this is a possible direction that we can go in terms of adding additional features to the solution. Then maybe the next one. Is it possible to extend the solution adding other relevant for our research public data sources? Martina, can you get this?
Sure. We do have an inventory of about 200 public data sets that we already support, and it works in a plug and play principle. They're all mapped to each other, and they're already ready to be used and plugged into the UI. They come also with automatic handling of updates. And in case the customer already have their own ontology model, we can also customize so that they work with their ontology model. We also have the option to expand the graph by using proprietary data, and there either the customer can do the mappings themselves, so we can also support them so that everything is integrated in the common ontology model.
Thank you. Another interesting question is, when the platform recommends a specific target as promising, how can the researcher be sure to trust? How can he or she trust this recommendation?
Maybe this one is again for me, yes. Yeah. We leverage different kinds of algorithms and scoring mechanisms so that we can make sure that the researcher can take the decision and have high confidence in it. One way to score it is that global confidence score that my colleague Ilian just mentioned about. It is based on RDF star, where we compute how this relationship that we have extracted is extracted, what's the provenness, and so on. Another mechanism is that we do have provenness and provenness scoring for all of the data that we provide and for all of the facts. Sometimes we also normalize scoring that comes from the public data source itself. One example here is StringDB. We normalize the score so that it can be compared to other data sources that we also integrate in one place. Yes, and for NLP, I already mentioned we do have the annotation scoring, and we do also normalize them so that they can be comparable to other facts as well.
Thank you. Another one is around the ability to load our own proprietary research data in the system. Is this possible?
I kind of answered it with a previous question. Yes, we can also integrate proprietary data. We can also assist the customer in doing so. We can also help the customers implement their own custom ontology model in case they have it as well, and to integrate all of their data seamlessly with the data that we extract from NLP and also from the public structured data sets.
Thank you. I think we already touched upon this, but still to make it like clear about that, does your platform support running machine learning algorithms on top of the knowledge graph? Apart
yes,
from the ones that we already have already present. I guess this is maybe the spin in the question. But please feel free to share your point.
Yes, I just wanted to say that yes, algorithms can be implemented. The exact mechanism of implementation can be done per algorithm, so it depends on the ML algorithm itself. So it's something that is more custom.
Yeah. And then a question about licensing. Is this tool licensed separately from GraphDB? Yes, this is a separate actually product that has GraphDB incorporated in it, but it comes with a single license that covers everything in terms of components. So there is simply no need to have GraphDB separately licensed if you purchase a license for the target discovery solution we just presented. And maybe the last question, because we're approaching the end of the session, what are the biggest advantages of the combination between knowledge graphs and AI? Maybe Martina, your take on that.
Yeah, I think you presented it on one of the slides already, but the knowledge graph is really a place where we can store all of these public data sets in a normalized way, and we can easily traverse through this whole graph, and we can easily get from one entity type to another entity type without having to switch to, let's say, other products. Also, a knowledge graph can help us to find new facts by utilizing the so-called inference. By combining data from different data sets, we can find out new facts that haven't been mentioned before in any of them. And of course, NLP with the knowledge graph is a great combination because of the semantics, but not only. And we do have seamless integration between data extracted from the NLP. It flows to the knowledge graph, and the NLP, let's say, dictionaries that we use, they're so-called gazetteers, are normalized to use data that comes from the graph. Maybe something else that you would like to mention here?
No, you put it quite nicely. It's indeed the ability to have all relevant data interlinked in a single layer, so that you can have an application like the one that we presented here, consuming all those insights specific to your use case and helping your researchers to actually be much more efficient when it comes to deriving findings on top of both public data, but also your proprietary data, everything consolidated against a specific schema and use case. This is really important. Just checking. I think we have a last question, and we will be maybe finishing after that. Approximate search. When no candidates find, is there a way to make the search approximately? This is an interesting one.
maybe I can answer
Go
that.
ahead.
So yeah, if we relate to the semantic search, of course you can search with a string instead of an exact entity. Even if you search for the exact entity, you can still get related objects. Like if you search for a gene that's called MGMT, then you will find also other genes that are called in a similar way. Especially for gene symbols, you can have some ambiguity. With KRAS or other sorts of RAS proteins, it's a huge family, so that's one example. Another thing is that we do have an option called more like this. This means that in the dashboard itself, you can put a widget that provides all similar candidates to the one that you're currently exploring. So if you have MGMT, for example, you will get in similar other methyltransferases, for example, or other proteins that have something to do with glioblastoma, or others that have something to do with DNA modifications. You can also customize how this works. And we're also working in other types of searches that would allow you to perform, for example, Boolean searches and perform more powerful searches than the one you saw today. So definitely, if you're interested in learning more about it, we can definitely show you something into more detail.
Thanks, Martina. I think we better call it a day. We are beyond the hour. Thanks to everyone for making this discussion really interactive one with sharing all your questions. Thanks also for participating in the poll that we broadcasted during our presentation. Yasin, would you like to have some concluding words?
Sure, yeah. Thank you both very much. It was indeed a very interesting presentation. Like I said, this was recorded, and to everyone interested, I will share the recording tomorrow, so you will have it available, so you can watch it again if you need it, or share it to somebody else. And with that, I'd like to end the webinar, and thank you again all for attending, and see you on the next one. Goodbye and have a nice day or evening.